Our next speaker is Holly Ganz, who is the founder and CSO of AnimalBiome. And as I mentioned, she will tell us about incorporating PacBio technology into her product to help diagnose what might be wrong with your cat or dog.
Good afternoon, everyone. This seems quite different from the ocean work, but I can see some overlaps, actually, in terms of building reference databases. So this afternoon, I'm going to talk to you about assigning species-level taxonomy to full-length 16S ribosomal RNA gene sequences using a pipeline that we've developed called FlexTax, which is a hybrid approach to classification for bacterial species. So I'm talking about the other genome that you may not be thinking about in your animals, but the microbiome is also matters for animals, and I guess for plants too. So AnimalBiome is a company that grew out of research I was doing at UC Davis, and I launched a citizen science project to try and let people be aware of the microbiome and why it might matter to them, because science funding was getting harder to get. And all these people came to me and said their cat or dog had terrible diarrhea and they needed help. So I created an at-home testing platform where people could send in their poop samples to us and we would sequence it. And then we realized that a lot of them were dysbiotic and that there might be ways that we could help these animals and their owners by creating supplements to support gut health. We transitioned recently, a couple years ago, to using the PacBio Sequel IIe, and it has tremendously advanced the data that we're collecting, and we're very, very pleased with it. So this is where I think this is kind of similar for why do you want to have good reference genomes? So long-read sequencing platforms have enabled full-length gene sequencing for this marker gene, 16S ribosomal RNA, and it allows us to get many sequences that can be classified, and ideally we were wanting to classify them to the species level. We don't really expect strain level with this amount of sequencing, but in the future, so we can do more and more, we would love to get that as well. Just as a refresher, this gene is about 1,500 base pairs and has 9 variable regions and 9 hypervariable regions. And so this was allowed Carl Woese and others to develop it as a wonderful biomarker for bacterial diversity. And the commonly sequenced regions are either these V3, V4, and V4 that were used a lot during using short read sequencing. But long read sequencing has allowed us to do the whole thing, so V1 through V9, and even beyond. The problem is that there's sort of twofold problems. One is that the current pipelines that are used for processing these data were developed for shorter amplicons, so they don't perform that well with the long reads. And a second problem, which I should have probably mentioned here, is the reference databases have a lot of short sequences in them, and so they're not long enough for us to allow us to take advantage of this increased data. It's important, though, that we do this because the replacement of higher level classifications like genus or family with species level assignments is really valuable and allows us to identify pathogens and other species that might look the same at a higher level. And here's just one example with Streptococcus. Streptococcus is a group that includes pathogens, commensals, as well as probiotic species. And if you're just sequencing V4, all you're going to see is that it's Streptococcus. Meanwhile, there are many, many species, and they differ a lot. So here's our proposed solution, FlexTax. It's a hybrid approach that utilizes both Chimes2's sklearn or scikit-learn classifier as well as vSearch to do taxonomic classification of these full-length 16S ribosomal RNA gene amplicon sequences. And this work really was inspired and built on PacBio's pipeline, their HiFi 16S workflow, which is available on GitHub. Their pipeline combines vSearch, a naive Bayes classifier, and multiple rounds of taxonomic assignment against 3 different reference databases, and is able to assign species-level taxonomy to many of the input sequences. A challenge, though, with this approach is that the use of multiple reference databases requires the end user to then reconcile taxonomies that don't agree with each other at the end, and it's very messy to interpret those And what that really points to is that the reference databases need to be cleaned up. So this is just showing their workflow, and we still benefit from many of these components, looking at like Data2 and Chime. I'm just going to highlight here that we took the concept that they had here of doing multiple rounds of taxonomic assignment, but we're using a single reference database instead. And this is key difference is sort of highlighted in the starred box in this workflow. So FlexTax is a hybrid approach that utilizes both the Chime2 scikit-learn and vSearch classifiers to assign taxonomy to full-length 16S ribosomal gene amplicon sequences, and it's available on GitHub at AnimalBiome's FlexTax. And so what are the benefits? So it allows us to benefit from this relative strengths of 2 good classifiers. It allows us to have flexible thresholds and parameters to accommodate a diversity of sample types and needs. And it employs a single consistent reference taxonomy, which with the caveat, there's continual work that needs to be done to continue to add and improve this reference database. Here's a flow chart for FlexTax showing the processes of database curation, HiFi read processing, taxonomy assignment, and then the merging of the taxonomic assignments. So a big part of this work was that we downloaded the Silva database and trimmed it to full-length or nearly full-length sequences of this gene. We removed species-level taxa that had terrible names like metagenome, whatever, or there's lots of junk in the reference databases that just need to get culled. We removed taxa that didn't belong there. And then we added important core taxa that we have been generating genomes for from our own efforts to isolate bacteria associated with cats and dogs. And so I'm going to just show you a few examples of how we evaluated the performance on mock microbial communities, different mock microbial communities made by Zymo, and then also with microbiome samples that were collected from cats and dogs. So Zymo has 2, the Zymobiotics Microbial Community Standard and also their Gut Microbiome Standard. And as I'll show you, we found that FlexTax outperformed sklearn and vSearch individually in approximating the taxonomic composition of these 2 reference standards. So this is a graph showing the relative abundance of different bacteria. And I don't think I can point, right? So starting with Bacillus and then going down to Staphylococcus staph aureus. And then we have the classification coming from sklearn, vSearch, and then FlexTax going from left to right. So sklearn failed to provide species-level resolution for many of the sequences, including some really well-known ones, including Bacillus subtilis, E. coli, Salmonella enterica, and Staph aureus, which it only was able to classify to the genus level. vSearch performed better and in fact was similar to FlexTax, which is our hybrid approach for this community standard. And so I'll just jump to FlexTax. It accurately quantified the relative abundances of Listeria, Enterococcus faecalis, Salmonella enterica, and Pseudomonas aeruginosa. It did slightly overestimate Staph aureus, and it slightly underestimated Lactobacillus fermentum. And it did detect Bacillus subtilis and E. coli, but not all of the sequences received species-level assignment, so sometimes some of the reads were going into the genus level. We think that with additional manual curation and correcting more taxonomic mismatches, we may be able to help resolve this issue, so this is still a work in progress. There are a lot of different bacteria, so it's going to be ongoing. And then we also looked at how it performed with this other Zymobiotics gut microbiome standard. So this is a standard that requires that you extract the DNA, so it's going to reveal biases that might arise from your DNA extraction method and other variations that can be introduced from different lab processes. So none of these classifiers actually recovered the expected number of sequences for the bacterial species in this standard, and this may be due to technical variation that was introduced with the creation of the standard, our DNA extraction methods, our sequencing library prep, or maybe due to limitations of our reference database, because it has different taxa in it than the prior microbiome standard. Nonetheless, I want to show you the results we got with sklearn, vSearch, and FlexTax again, with the Zymo control on the right. So FlexTax accurately quantified the abundances of Enterococcus faecalis, C. diff, Fusobacterium nucleatum, Salmonella enterica, and Akkermansia mucinophila, with the observed absolute relative abundance values closely matching the expected values. It did overestimate Bacteroides fragilis and Prevotella corporis. It underestimated E. coli slightly, and Faecalibacterium pronouncii. And it misclassified Veillonella rugosi as a different species of Veillonella. On the other hand, sklearn was unable to confidently assign species-level classifications for such well-known groups as C. diff, E. coli, and some Veillonella, and defaulted to genus-level calls. And for vSearch, it had more species-level assignments than sklearn, but it left about 20% of the reads unclassified. And this is probably due to the stringent thresholds that are set with that classifier to ensure higher accuracy. So then we also wanted to assess how it performed with microbiome samples, recognizing that we don't know the truth here. We don't know what the composition really is, unlike the microbiome standards. And what we found here when we look at the proportion of the reads that received a species level, these are the amplified sequence variants that received a species level assignment. So this is different graphs looking at the Violin plots with the sklearn in red, the vSearch in green, and FlexTax in blue. We see that FlexTax performed better than sklearn and vSearch. And I should also say we have cat gut on the top left and dog gut on the bottom left. And then oral samples, cat and dog, on the right. And the blue, we can see we got more assignments. So it did perform best of the three. And where we had 65 to 76% assigned at the species level versus sklearn at 63 to 71%, and vSearch performed much worse at 29 to 39%. Another thing to note here is that we get better assignments for fecal samples than we do for oral samples. And this is not surprising. The gut microbiome has been the subject of a lot more research than the oral microbiomes. Likewise, we get more assignments for dog samples than cat samples. And we also, if I would add in humans, we would do better for humans than dogs, than cats, and then, than other species that are less well characterized. So that is to be expected. So then I wanted to just tell you about 3 recent studies that we published using the FlexTax pipeline. One of them was looking at this, the microbiome in healthy dogs. And it's our first time publishing, actually it's the first paper publishing species level characterization of the dog microbiome using this technology. We also did a study looking at the oral microbiome across oral sites in cats who are healthy, had periodontal disease, or chronic gingiva stomatitis. And we used this approach to look at bacterial engraftment following the delivery of oral fecal microbiota transplantation capsules to dogs who suffer from a chronic enteropathy, or it's more commonly referred to as IBD. So because we do microbiome testing for consumers and we let them assess what their cat or dog's microbiome looks like to try and help them understand what might be going on when they're having a chronic GI condition, it's important for us to understand what the healthy microbiome should look like. And we all know there's a lot of variation among individuals, so it's not a trivial thing. But there is this concept of the core microbiome where we try to look at what are the taxa that are found in most individuals. And so in this paper, we describe that. We give statistical ranges for each of the taxa that were prevalent in this population of healthy dogs. And we did find the brown on sort of that's increasing up on the right are non-core species. So there's a lot of variation, and that's what that's sort of showing, that not everything is in the core microbiome. And then we also recently did a study where we were looking at the oral microbiome across different sites, looking at the biogeography of the mouth in cats who are both healthy as well as various disease states. I'm just showing you the healthy cats here. And so there's differences in different parts of the mouth. And then we also found, by being able to do species level differentiation as opposed to just looking at V4, we were able to identify 21 species that were enriched in healthy cats compared to cats who had chronic gingiva stomatitis, which is a really terrible inflammatory condition that's very painful. And the main treatment today is to extract all of their teeth, which works, but it's kind of not ideal. What we found was that condition is associated with an absence of these organisms, as opposed to an enrichment of some pathogen. And there was only one group, Flexinella, that was elevated in the cats with FCGS. And then we also collaborated on a study with the University of Helsinki School of Veterinary Medicine, where they recruited dogs with chronic enteropathy who that was antibiotic responsive. So they were giving them Tylosin, but they were having to take Tylosin every day. And I think we all know, it's not really an ideal solution. It'd be nice to find some other approach. So they were exploring the question of whether delivery of an oral fecal transplant capsule could reduce the relapse rate when they went off antibiotics. And here we looked at what are the bacterial taxa that engrafted in the recipient following FMT. And as you can imagine, the more specificity we can get, the better we're able to really assign it as having been something that came from the host to the recipient. So species level is critical. In the future, probably we'll be looking at strain level differences to better be able to identify that. And likewise, I guess I'll add that if we can't, our ability to detect this depends on our ability to differentiate between the different bacteria and assign and identify that it came from the donor. And so if the recipient has something that looks broadly similar, we won't be able to tell that it came from the donor. And so in conclusion, we've developed a pipeline, FlexTax, that we welcome everyone to use. And we have a tutorial. We welcome your questions. Happy to help. And that this pipeline, we are finding results in improved taxonomic assignments. This in large part is, I think, due to curation of the reference database to allow us to improve the taxonomic assignments. And we tested it in the field in cats and dogs and found that the species level assignments perform better in fecal versus oral samples, as well as in dog versus cat samples. And while this is something that we'll be continually working to improve, we believe that the performance we're finding so far across these different host species and microbiome types demonstrates its adaptability to a range of different applications. And we have a wonderful team who's very committed, and they are really loving being able to do our own sequencing and generate these nice data. And I want to thank you for your attention, and I'll take questions at the PacBio booth or wherever we're doing that later. Thank you.